The rapid development of biotechnology has increased the possibilities and efficiency of selection. Cell engineering methods allow to accelerate the rate of improvement of breeding in commercial herds, create valuable animals with productive traits, clone them, quickly get record holders and whole herds with record milk yield. We have divided the video into three main parts: methods of creating transgenic animals, animal cloning methods, and methods for creating chimeric animals.
Methods of creating transgenic animals. Animals with introduced forage gens are called transgenic. Transgenic animals can be created by introducing DNA from another organism. Take mice for example. DNA can be injected into mice. 1. The use of viruses capable of introducing forage DNA. Infection of shells is carried out at the early stage of embryo development, after which it is transferred to a female mouse. 2. Macroinjection into the nucleus of the male reproductive shell, sperm, at the stage of the fusion with the female reproductive shell, ovum. 3. Introduction of modified stem cells, immature shells, at an early stage of embryonic development before implantation of the female mouse.
Establishing transgenic mice with retroviral vectors. A class of viruses are called retrovirus because they do anything the other way around. The concept of retro is due to the fact that retroviruses have two strains of RNA in their genome, which they convert into DNA. Then they transfer this DNA into the cell nucleus and insert it into the host genome. Thus, the retrovirus genome becomes part of the host genome and the cell can never get rid of it. A vector is a safe genetic construct that doesn't cause disease. Cleavage stage embryos, usually at the 8-cell stage, are infected with an effective retrovirus carrying a transgene. Implanted females foster mother give birth to transgenic pups. Mating are carried out to determine which pup have the transgenic in their germ-line cells. Transgenic lines can be established from these founder transgenic animals. Disadvantage of retroviral vectors. Vector can transfer only small fragments of DNA. It's absolutely necessary that there should not be any retroviral contamination in which either a product is to be synthesized by the transgenic organism or the transgenic organism is to be used as food. Transgene introduced on some retroviral vectors are silenced.
Establishing transgenic mice by DNA microinjection. Because of the disadvantages of the retroviral vector method, microinjection of DNA is currently the preferred method for producing transgenic mice. Microinjection is a method of injecting DNA with a microscopic glass pipette capable of accurately penetrating the cell wall and membrane. Even the nuclear membrane is necessary. Step 1. Eggs are obtained from donor females and then mated with males. Step 2. Purified samples of the transgenic construct are microinjected into the male pronucleus of a fertilized egg. Step 3. Implanted female gives birth to transgenic pups. In microinjection method, the injected DNA integrates at random sites within the genome. Therefore, not all of the transgenic pups will have their appropriate characteristic. Need to check mouse pups for DNA using various biotechnological methods. The gene may not be active due to the site of its introduction or being overly active, which can interfere with the normal functioning of the body.
Establishing transgenic mice with genetically engineered embryonic stem cells. Stem cells are obtained in the early stages of embryonic development. On days 5 or 6, the embryo consists of several tens or hundreds of cells. This is the blastocyst stage. The transgene is introduced into the stem cells. These cells are grown in a special medium and then inserted into blastocyst into foster mothers. Transgenic lines can be established by crosses from founder mice that carry the transgene. Gene knockout is a method in which a selected gene is removed from the body or turned off. Stem cells can be used to create knockout mutants. Step 1. Get the embryonic stem cells. Step 2. Genetically engineer the embryonic stem cells. This method is characterized by the concept of homologous DNA recombination. This is the exchange of regions between two similar or identical chromosomes. Homologous recombination was the main method for causing a gene knockout. This method involves creating a DNA construct containing the desired mutation. For knockout purposes, this typically involves a drug resistance gene in place of the desired knockout gene. The construct will also contain a minimum of 2000 base pairs of homology to the target sequence. The construct can be delivered to stem cells either through microinjection or electroporation. This method then relies on the cell's own repair mechanisms to recombine the DNA construct into the existing DNA.
Knockout cells are selected by culturing them on antibiotic media. Only those cells that have resistance gene survive. One of the aims of targeted gene disruption, gene knockout, is to determine the consequence of inactivating. A gene-turned off organism can be used to study human disease. Cloning. Reproductive cloning could allow researchers to clone animals with potential benefits for the fields of medicine and agriculture. Another reason for cloning is that there are populations of animals that are on the verge of extinction. Creation of animals capable of producing proteins usual for their species is especially urgent. It was reported about development aimed at obtaining pigs capable of producing human interferon.
Reproductive cloning. In reproductive cloning, researchers remove a major somatic cell, such as a skin cell, from an animal that they wish to copy. They then transfer the DNA of the donor animal somatic cell into an egg cell that has had its own DNA-containing nucleus removed. Researchers can add the DNA from the somatic cell to the empty egg in two different ways. In the first method, they remove the DNA-containing nucleus of the somatic cell with a needle and inject it into the empty egg. In the second approach, they use an electrical current to fuse the entire somatic cell with the empty egg. In both processes, the egg is allowed to develop into an early-stage embryo in the test tube, and then is implanted into the womb of an adult female animal. The adult female gives birth to an animal that has the same genetic makeup as the animal that donates the somatic cell. This young animal is referred to as a clone. Reproductive cloning may require the use of a surrogate mother.
History of cloning animals. The history of cloning began a long time ago from the beginning of the discovery of reproductive cells. 1826. Carl Ernst von Baer was an Estonian biologist who discovered the mammalian ovum, the reproductive egg in female mammals. He made significant contributions to the study of the embryonic development of animals. 1833. Oscar Hertwig proved that fertilization occurs due to the fusion of a sperm and egg cells by studying sea urchins. 1958. Josh Carden successfully cloned a frog. 1985. Boris Viprintsev cloned a mouse from an embryonic cells using electrostimulated cell fusion. 1996. Dolly was cloned by Kate Campbell, Ian Wilmot and colleagues at the Roslin Institute. Dolly was born on 5 July and had 3 mothers. What about human cloning? The possibility of human cloning has raised controversies, as ethical concerns have prompted several nations to pass legislation regarding human cloning and its legality. As of right now, scientists have no intention of trying to clone.
40 years, scientists have conducted cloning experiments in a wide range of animals using a variety of techniques. In 1969, researchers produced the first genetically identical mice. Shortly after that, researchers produced the first genetically identical cows, sheep, and chicken by transferring the nucleus of shell, taken from an earlier embryo into an egg that had been emptied of its nucleus. It was not until 1996, however, that researchers succeeded in cloning the first mammal from a mature shell taken from an adult animal. After 276 attempts, Scottish researchers finally produced Dolly, the lamb from the other shell of a 6-year-old sheep. Besides cattle and sheep, other mammals that have been cloned from somatic shells include cat, deer, dog, horse, mule, ox, rabbit and rat.
What are pros and cons of cloning? Pros. At first, it allows to understand genetic research. Then, cloning using one's cells enables one to quickly recover from traumatic injury. Then, cloning allows replacement of damaged organs. You don't have to wait for donation on a failing organ, thus saving a life. This technology allows individuals to produce a clone of themselves. You can choose the trait you need for your genetically created child. Cloning is not only clone humans, but can also clone plants and animal species, offering a lot of benefits. Using the genetic material of dead animal species help in expanding the diversity of the genes. Cloning plants aid in altering the gene, thus making them resistant to disease. Cons. At first, it's against God creation. Creating human life, though cloning is against God's wishes. The genetically created person doesn't have feeling or empathy. Then security threat. Your skin cells can be used to clone you without your knowledge, and it will be difficult to defend your rights as the true human being. Explosion. Cloning of body organs not only extend human life, but also can lead to a lot of malpractice where undesired traits may be reproduced for vested interests. The cloned person adopts the original person's age, resulting in faster aging. It will also lead to a lot of identical individuals in the world. Cloning creates an artificial person, and it interferes with the natural process of procreation and reduces the value of human life.
Chimeric animals. The essence of the method of obtaining chimeras is the artificial union of embryonic cells of two or more animals. Animals can be either of the same breed or of different breeds and even of different species.
Methods for obtaining chimeras. Several methods are used to create chimeras. First, aggregation is the process of combining. Aggregation uses the embryos of different animals that have reached stage of 8 blastomeres. Blastomeres are one of the stages of embryo cleavage. At the stage of the third division of the zygotes, 8 blastomeres are formed. These blastomeres are placed in special environments that facilitate their fusion. The result is a 16-cell embryo from different blastomeres. Composite embryos develop in vitro to blastocyst stage, after which they are implanted into the uterus foster mother, where they continue to develop. Second, injection. This method used embryos at the stage blastocyst and used micromanipulators to inject donor cells into blasted cell of the recipient embryo. The purpose of experiments on mixing animal and human cells is to grow donor organs, the problem with the lack of which is very acute today. Chimeric animals are used to address problems of genetics, embryology, biomedical research, and may of interest to livestock practices.
Modern cell engineering is the clearest example of the unity of science and practice. After all, it allows you to experiment with cells, not with whole organisms. The latter is much more difficult and sometimes impossible, especially in the case of mammalian animals and humans, or when obtaining distant hybrids. We have tried to show how research, which until recently appeared to be purely theoretical, in a few years gets a real practical output for the benefit of the health of present and future generations. Currently, cell engineering is the most promising and harmoniously developing field of biotechnology. The present methods of cell engineering are associated with success in various fields of science, medicine, pharmacology, production, and agriculture. And what is important? The use of cell cultures allows us to overcome many problems of bioethics associated with the use of animals in scientific research. That's about it.
We have tried to show how research, which until recently appeared to be purely theoretical, in a few years gets a real practical output for the benefit of the health of present and future generations.